Welcome back to NPTEL online course on Introduction to Complex Biological System. During our last lecture, we were discussing about our innate immune response. Today, I will mostly focus on our adaptive immune response. So here, I will cover the basic concept of adaptive immunity, followed by like different components, some cell or some molecules which are involved in adaptive immunity. Those I will discuss in detail. And finally, I will explain the mechanism of adaptive immune response. So now, going into the adaptive immunity, so this is the precise and powerful defense mechanism of host. So when I was discussing about innate immune response during the last lecture, I specifically mentioned that during the innate response, at some stage when particularly dendritic cell and macrophage, they are engulfing pathogen and they are processing the information. That means when they are those pathogen they are getting inside this dendritic cell, they will break down those pathogen. You can consider some bacteria, and it will make small protein fragments called peptide, and those peptide will be presented on the surface of this antigen presenting cell like dendritic cells. Now this dendritic cell and macrophage, although they play very important role in innate immune response, but they are initiating the adaptive immune response. So when they are presenting antigen on their surface, now they will guide other cell, for example T cell, to develop our adaptive immune response. So I will discuss the detailed mechanism today. Although this adaptive immune response overall it's a very complex thing, and within just 20-30 minutes it is really difficult to explain all the pathways and mechanism involved there, but I will highlight the basic concept, the basic mechanism of this adaptive immune response, and at the end you can appreciate the complexity of a biological system, particularly in this context, in the context of our adaptive immune response. So the adaptive arm of our immune system can better recognize the pathogen or the enemy, you can say, and it can eliminate the enemy, and finally or more importantly, it can remember the invading pathogen. So in near future, if we get again attacked by the same enemy, same pathogen, so our adaptive immune response will act very promptly. Now the second point is the development is dependent upon earlier innate pathways. As I just mentioned, that at the beginning innate immune system, the innate response is going on properly, and it is actually helping to develop immune response, the adaptive immune response against that particular pathogen. And adaptive immunity provides a second and more comprehensive line of defense based on the struggle of innate immunity. As I mentioned during the last class, that innate immunity we are referring as first line of defense, and that's why we are saying that this is second and more comprehensive line of defense mechanism of host. Let's see what are the different components of adaptive immune system they are present and how they are playing important role. So now I divided those things in two different categories here. So the cellular components, as you can see here, and the molecular components. So the first thing I will concentrate on antigen presenting cells. So professional antigen presenting cell, I would say the first thing come into our mind is dendritic cell. Dendritic cell. From now, I will refer these cells as just DC, dendritic cell. And another one is macrophage. Okay. So they are professional antigen presenting cell. And the molecular components means it is associated with this antigen presenting cell, that is called major histocompatibility complex. It's very important. So those are some surface protein produced by this antigen presenting cell, and they present the antigen, the peptide antigen particularly, on the surface of this antigen presenting cells. And what peptide? So in this context, whatever I am discussing now, I would say the peptide coming from the pathogen. Sometimes endogenous peptide can be presented in a different context. I will touch that part also sometime later. But now consider just a peptide coming from some foreign body, like it can be some I would say some bacteria or some virus, something like that. Now this MHC you can broadly classify like two types of MHC present. One is called MHC class 1 and MHC class 2. So this is class 1 and this is class 2 MHC. Okay. Now this class 1 MHC is present in our body in all nucleated cells, not just antigen presenting cell. They are present in all nucleated cell. This is very important information. But MHC class 2 is present on antigen presenting cell, particularly professional antigen presenting cell. And what is the other difference? As you can see here, their domain organization, their structural features is little bit different between class 1 and class 2 MHC. As you can see, the class 1 MHC that cross the membrane, so this is the plasma membrane, okay? Plasma membrane. Same here, this is also the plasma membrane. Now, if you see class 1 MHC, they cross the membrane just once here, but the class 2 MHC, they cross the membrane twice. And because of that, their domain organization is also different. So in class 1 MHC, we say this is our heavy chain, and this is called beta 2 microglobulin or beta 2M. Sometimes we just say beta 2M, beta 2 microglobulin. So the heavy chain is just single polypeptide, and the beta 2M is another polypeptide. So this is some kind of example of, I would say, quaternary structure of protein. Two different polypeptides, they are associated together and they are forming the complex. But here the most important thing is, on top of this heavy chain, I would say in this in this MHC, you have some peptide binding group or cleft. So the peptide will be present here. So this is the antigenic peptide. This is the peptide presented by some MHC molecule. Okay, and later on, this peptide loaded MHC will be recognized by some T cell receptor. I will discuss soon. But in case of class 2 MHC, more or less it's kind of similar. Only here, as you can see, alpha chain and beta chain, the organization is little different, and also the peptide binding cleft is also little bit different. Although in this figure you cannot differentiate, but I would like to say that in case of class 1 MHC, the peptide length is generally from 8 to 11 amino acid long. Generally, 8 to 11 amino acid long antigen can be presented by class 1 MHC. But in case of class 2 MHC, the length can be even bigger. So it is possible. That is because of some in-depth structural features, how the antigen is being presented. I am not going into that detail in this, in this lecture. Now, as you can see here, the T cells. So what I particularly mention if I summarize here what is happening, so that here if I say this is our antigen presenting cell, you can say this is DC. Now, when it endocytoses some bacteria, okay, this is bacteria. In the next step, this bacteria will be broken down into small pieces and it will be presented on the surface of this dendritic cell or APC, antigen presenting cell, by MHC molecule. This is MHC molecule. Okay. Now, this dendritic cell, they can express both class 1 as well as class 2 MHC because they are professional antigen presenting cell. Now, what will happen? T cell here, if this is our T cell, on the T cell you have another receptor called TCR. This is TCR, T cell receptor. It will recognize the peptide loaded MHC on the surface of antigen presenting cell, and therefore T cell will differentiate and some other activities will be happening. Now, let us see how TCR looks like. So, here this is the T cell receptor. So, as you can see in TCR you have alpha chain and beta chain, and this TCR you have the variable region and constant region. So, this variable region, constant region, all those module, all this domain, they have some kind of some kind of protein fold which is called immunoglobulin fold, which I will be discussing in the next lecture when I will discuss in detail about the antibody structure. But for the time being, just try to understand their overall structural features is same, that is called IG fold. So, this is one IG fold, this is another IG fold, another IG fold. So, that way they are organized. But that is not important here. The important thing here is this TCR here, this is the binding portion. So, through this portion they can recognize the peptide loaded MHC on the surface of APC. But here we have to be careful about our understanding that this TCR on the T cell surface, this TCR, it can reorganize themselves. Their sequence, it can vary. So, based on the TCR can just like antibody, as many of you know that antibody, like the sequence and the specific structure, the antigen binding site particularly, it can vary a lot, like huge amount of diversity available. Similarly, in the TCR region also this diversity exists. So, as a result of that, they can recognize peptide loaded MHC. And this MHC, they have some kind of restriction, that MHC are restricted so that this MHC cannot interact with any type of TCR. There are some kind of matching is required. So, this is we say that peptide loaded MHC will be recognized by cognate receptor, a specific type of T cell receptor, right. So, that is all. Now, what we discuss here? We discuss about the antigen presenting cell, and they present antigen through MHC molecule, and also we discuss T cell, which they have T cell receptor on their surface, and this TCR and this MHC, this recognition, it is, I would say, one of the very important step in the adaptive immune response. Now, the next thing is coming here is B cells. B cell is also very important. Like T cell and B cell, they are the major thing in our adaptive immune response. As you can see, B cell. So, this B cell, on their surface, they have another receptor which is called B cell receptor. So, see, so this structure, if you see, this is almost look like an antibody. This is an antibody. But what is the difference between the BCR and an antibody? That BCR means B cell receptor, they are membrane bound immunoglobulin. So, as a result of that, I would say if this is B cell, so they have this here, but they are still bound to the plasma membrane. So during that time, we are saying that this is our B cell receptor. But this B cell, when it will differentiate into plasma cell, they will secrete a specific type of antibody from those cells. Those proteins are now being secreted, and we will say those are antibody. Now then, what is the role of BCR? So BCR present on B cell surface, they also recognize antigen, but that antigen, they don't need to be presented by MHC molecule. That is the very important thing. I would say, if this is B cell and you have here BCR, they can recognize antigen, and this antigen might not be a small peptide, it can be a bigger protein also. So as a result of that, what will happen? The B cell can directly recognize antigen, and it can be a some protein, secretory protein coming from some pathogen, from bacteria, and it will be recognized by this B cell receptor. So this is the difference between the recognition of antigen by B cell and T cell. TCR need the help of MHC present on antigen presenting cell, and the antigen presenting cell processing that protein, the foreign protein, into small pieces, and that peptide is being presented, and now this peptide loaded MHC can be recognized by T cell receptor, but in this case directly B cell can bind to the antigen. That's all. Now I would like to mention here this immune system, particularly this related to adaptive immune response. It's huge advancement in our biomedical field. If you see here, particularly I am mentioning few of those Nobel Prize related to our immune response, particularly related to adaptive immune response also. I'm just mentioning, I'm not bringing a scientist name here, it will be a very big list. So here, as you can see, in 2000, so all are in the category of physiology and medicine. So in 2011, I would say discovery of dendritic cell and its role in adaptive immunity. So with this award, maybe some other other findings by other scientists are also involved there. That means that prize when awarded, is clubbed together with some other finding, but I am only focusing on related to our adaptive immune response. Now if I just go back, like I started with 2011, now 1996, I would say the role of MHC TCR interaction in adaptive immunity. It has been awarded, the Nobel Prize has been awarded. And in 1987, the genetic basis of diversity of antibody produced from B cells. I just mentioned that B cell can produce countless different types of antibody, right? And based on the pathogen. So that, the diversity of antibody, that particular genetic basis, it has been discovered. And in 1984, the production of monoclonal antibodies, it has huge therapeutic, therapeutic importance also. And then in 1980, the discovery of MHC molecule, the major histocompatibility complex. And 1972, the chemical structure of antibody. As you can see, all those major keyword I already mentioned, like MHC, TCR, antibody, all those things I mentioned, dendritic cell, adaptive immunity, innate immunity, right? So let's see, now I will discuss the mechanism of adaptive immune response together. As I already mentioned, it is a complex process, but I will try that as simply as possible. I would like to explain that. Let's see what happened. I will go step by step. Now, whatever I mentioned when I was discussing about cell and components of adaptive immune response, now I will add some additional complexity here, okay? So, from here, if I start, this is dendritic cell, and it is presenting the antigen. So, this is antigen. I would say here I am writing peptide loaded, little bit mistake, peptide loaded MHC, peptide loaded MHC. Now, T cell you have here, right? T cell. T cell, now we have TCR. This is called TCR or T cell receptor. So, a specific interaction between TCR and MHC will lead this adaptive immune response. Now, we have like majorly, there are many complex things there also, but majorly we have two different types of T cell involved here. One is called CD8 T cell. That means in this T cell, beside the TCR, you have another co-receptor. This is called co-receptor. Okay. What kind of co-receptor? CD8 co-receptor. The major receptor here is the TCR and this is the co-receptor. Now, when this interaction is going on, MHC and TCR of CD8 T cell, then this T cell will differentiate into cytotoxic CD8 T cell. Okay? So, we will say cytotoxic CD8 T cell. Now, they have the similar TCR. The TCR, they saw this antigen which was loaded on MHC. So, as a result of that, you have TCR present here in this cytotoxic CD8 T cell. And next, what will happen? From the name, it suggests that cytotoxic. So, as a result of that, this cytotoxic T cell, it can directly kill virus infected cell, like our host cell, in fact. So, the thing is, I would say, what happens if I say this is our virus infected host cell, right? Virus infected host cell. Now, the viral proteins are also present inside this cell, and finally, class 1 MHC will present antigen here, which is coming from this virus. They will be presented by class 1 MHC. And now this CD8 T cell, they will recognize this antigen because they already know, because they have already seen this antigen when it was presented on DC or dendritic cell. So, therefore, cytotoxic CD8 T cell exert its effect. So, from the name, it suggests that it will kill the cell. So, this CD8 T cell, it will kill the virus infected host cell. Okay, kill the virus infected cell. So, that way it will try to clear the infection. This I just mentioned about the CD8 T cell. But another group of T cells are available. So, when dendritic cell, so this is dendritic cell, when they are presenting antigen through class 2 MHC, so the previous one, whatever I discussed about the CD8 T cell and MHC interaction, This one I would say class 1. Okay. So class 1 MHC interact with the TCR of CD8 T cell. Now the class 2 MHC will interact with CD4 T cell. So here you have TCR and the co-receptor which is called here CD4 co-receptor. So now this CD4 T cell, when they will interact through antigen presenting cell via this MHC TCR interaction, this CD4 T cell, they will be differentiated, they will be existing as helper T cell. This is also called helper T cell, but they have this TCR present on their surface. Those are the TCR, T cell receptor. They know that what kind of antigen need to be, need to interact, right? Now this helper T cell, as their name suggests, they will not directly kill the infected cell. Rather, they will help some other cell, and that will be a very big thing in adaptive immunity. So this helper T cell will help B cell to produce antibody. It will help the B cell to be differentiated as plasma cells, and therefore that plasma cell will produce antibody. But how? What's the mechanism? Now what is going on here, like as I already mentioned before, that B cell, they can also directly see antigen that can directly bind to antigen through their receptor called BCR, B cell receptor. And as a result of that, what will happen, some of those antigen will be endocytosed. So antigen will come inside, maybe a big protein, it is coming inside, and it will be broken down into small pieces, and then B cell, they can express on their surface class 2 MHC. So this is class 2 MHC, okay? And they will be presenting this peptide on their surface. Now see what I told, that CD4 T cell, they already recognize the antigen loaded on class 2 MHC present on dendritic cell. Now this B cell, they are also presenting this, I would say, this class 2 MHC, and they are presenting this peptide antigen. Now this MHC class 2 presented on B cell surface, it will interact with the helper T cell. And as a result of that, this interaction will give the signal to B cell to be differentiated as this B cell. Now it will be converted into plasma cell. So that's why I am crossing this B cell. So no more B cell. I would say this is now plasma cell. So in this B cell, we had BCR on the surface. So this is BCR, and here in this cell, now they are secreting antibody. Antibody means some complex protein molecules, so that will bind to antigen. What kind of antigen? The antigen, the same antigen which was recognized by the BCR, the same antigen which is recognized by this I would say the class 2, the TCR recognize the antigen presented by the class 2 MHC on the surface of DC. But those antigens are coming from that particular pathogen, particular bacteria. As a result of that, this antibody will target the particular pathogen, say bacteria. Okay. So this way it will make a robust immune system, the adaptive immune system. In one way, CD8 T cell, they are directly killing some virus infected cell. On the other hand, CD4 T cells, after recognizing antigen on the surface of antigen presenting cell, they are helping B cell to produce specific weapon against that pathogen. Weapon means here antibody, and this antibody will mark those particular pathogen, and it will increase the efficiency of phagocytosis by some other cells, and also it will help in the killing process. It will be much better. So this is how adaptive immune system works. Now I have few things to discuss here, few points, because whatever I told, this is the basic thing, but still I am trying to bringing this all possibility, complexity also. Now here there are few points that what is the requirement of B cell by CD4 T cell, and then they are only getting converted into plasma cell and they are secreting antibody. They already saw antigen, so they can directly getting differentiated into plasma cell and they can secrete antibody. But there is a catch. So very important thing, because at some point, maybe in the first class of this immunology this module, I mentioned that T cells, although they are generated in bone marrow, but they are getting matured in thymus. When they are getting matured in thymus, those T cells, they are actually learning which is self and which is foreign. That means they are taught that these are the whole ensemble of our self antigen, and you shouldn't act against this antigen in future. So what I am trying to say, those T cells, they learn that what is self thing. So as a result of that, they shouldn't act against the self peptide. Self peptide means our host cell peptide. Otherwise, like a lot of different types of autoimmune disease will develop, right? But B cell, on the other hand, they are not taught this system. So they are matured in bone marrow in a confined environment, and they are not that way, they learn this thing. So as a result of that, this interaction between CD4 and this B cell through this antigen, like class 2 MHC loaded antigen and TCR, whatever I am showing here, this interaction is required because these CD4 cell or CD8 T cell, they are aware which is self peptide. So as a result of that, they will not act against the self peptide. So whenever this interaction is happening, that means the CD4 T cell is giving signal to B cell that you now can differentiate into plasma cell and make antibody against that particular antigen, and therefore it will make. Clear? So this is, I just mentioned about this B cell and T cell and their differences. Another important thing here I must mention that class 1 MHC and class 2 MHC. See, class 1 MHC, they present antigen when those antigen, they are processed or they are synthesized by our own machinery. As you already know, in the previous module I discussed that when virus infected host cells, virus, they don't have protein synthesizing machinery. They utilize the host cell machinery. So it's like endogenous protein. Although ultimately virus particle will be assembled, will be generated, but these peptides, since they are getting produced through endogenous pathway, our own cellular pathway, they will be presented by class 1 MHC. Similarly, some cancer antigen, for example, those are like, I would say again, some mutant protein, some bad protein which is produced inside our cell through endogenous manner. Those peptides will also be presented by class 1 MHC and which will be recognized by CD8 T cell, right? On the other hand, class 2 MHC, so that is actually present on antigen presenting cell, not in all cell. And this antigen presenting cell, they have very good capability to phagocytose thing. So as a result of that, they will phagocytose the pathogen. For example, some bacteria, it will be present now inside the phagosome, endosome kind of thing, and they will break those small pieces and it will be presented by the class 2 MHC. Now, if you notice here, these proteins are not being synthesized by the host cell itself. It is directly coming from the pathogen, and this is presented by class 2 MHC molecule. This is the difference. But here at the starting point, when the dendritic cell is presenting antigen to CD8 or CD4 T cell, because dendritic cell, they are professional antigen presenting cell, they have both class 1 and class 2 MHC molecule. They have ability to cross-present peptide. There are lot of interesting features available with dendritic cell. I am not going into that details, but all these things that help our adaptive immune system to either develop antibody against a particular pathogen, or they help CD8 T cell to become cytotoxic T cell and directly kill the virus infected cell or some other abnormal cell. Now, after all of those things, some cells will remain in circulation, remain in our body. Those are the memory cell, right? For example, some memory B cell. So in future infection with the same pathogen, they can quickly assess all those things and then accordingly they will behave. Here another important thing is where all those things are happening. I would say, like when T cells, they are not, they didn't see antigen yet, just like here, for example, like you can see they are seeing antigen loaded on the antigen presenting cell. So before that, those T cell we say naive T cell, okay? Naive T cell. Those naive T cell are present in different lymph node. For example, some like, particularly I would say spleen, for example. So now dendritic cell, it catches some information, I would say when it engulfs some pathogen. This dendritic cell has very good ability to move around. So it will migrate, it will go to some lymph node, some draining lymph node, and there T cells are waiting. So as a result of that, there they will teach T cell what should be done in the next step, okay? And on the other hand, macrophage generally present in tissue, like tissue resident macrophage. So their major role is there itself, but dendritic cell is very good at it. So it will travel through lymphatic system, and they will reach at some lymph node, and they will teach our naive T cell what to do next, okay? Now whatever I discuss, this is the summary. Now I will go very quickly, but it is already written here, step by step I will show. But whatever I just mentioned, it will be little bit less detailed here. So see, dendritic cell on the surface of MHC, like they present MHC on the surface, and then it can interact either with CD8 T cell or CD4 T cell through TCR, as I already mentioned. If the interaction is with CD8 T cell with the class 1 MHC loaded antigen, then that CD8 T cell will be now cytotoxic T cell. And now this cytotoxic T cell will recognize the virus infected cell or abnormal cell, as I just mentioned, for example, some cancerous cell also, with antigenic peptide loaded on class 1 MHC present on their surface. And the outcome is killing of the infected cell. It's very important. On the other hand, if you see, if the recognition between like class 2 MHC and CD4 T cell, then that is actually helper T cell, that CD4 T cell will be helper T cell, and they have that TCR on their surface. Now, I already mentioned how B cells see antigen and that antigen will be loaded on the surface of B cell by class 2 MHC. Don't forget it, this is not class 1 MHC, this is class 2 MHC, because this part we are talking about the interaction between the helper T cell and the B cell, and on the surface of helper T cell, whatever TCR you have, that will bind to class 2 MHC when it is presenting peptide, not the class 1. So that's why. Whenever this helper T cell will give signal to B cell, this B cell will be differentiated into plasma cell. This is very important step. And now this plasma cell will make antibody, and the final outcome is this antibody will bind to the target, and the neutralization and the induction of phagocytosis will happen. So as a result of that, like the infection will be cleared soon after all these steps. And as I already mentioned, some memory will be there also, and those memory cells will help at the end. And the end means, I would say, even after few months or few years after, like, if we are getting infected with the same pathogen. And that is all about adaptive immune response. Thank you very much. You can follow Genoa's immunology for additional details. That's all.